all. This is Dr. Mobeen from drbeen.com. Today we'll talk about Aviptadil. Aviptadil is a vasoactive intestinal peptide. So let's talk about this. This has been recently been discussed a lot about for the COVID-19, especially the pulmonary system effects plus the immune system effects. So let's start our discussion for Aviptadil. So I'm going to share my screen. And we'll go from here. So this is one of the articles here. So this article is about the vasoactive intestinal peptide is a neuropeptide produced by the lymphoid cells. So this is the basic peptide that we're going to talk about. So let's have a discussion. Look, Aviptadil itself is a synthetic, that means human-made, analog of vasoactive intestinal peptide. So it is an analog of VIP, and VIP is something that we produce in our body in general as well. So the VIP, the vasoactive intestinal peptide, is a 28 amino acids peptide. What does that mean? Our proteins in our body that are made, they are made up of amino acids. So amino acids are connected with each other through peptide bonds. And when they are connected with each other through peptide bonds, they make proteins. Normally, when 2 to 50 amino acids, they combine together to make a tiny short peptide or short protein, like here, then we call a small protein, we call it a peptide. If there are more than 50 amino acids that combine and make a bigger protein, then we call it simply protein. So protein and peptide are actually the same thing. Peptides are just shorter proteins. Now, VIP, vasoactive intestinal peptide, is a peptide hormone. It is produced in our body. It is a protein hormone. It is produced naturally in our body. Number one, in here, this is the hypothalamic suprachiasmatic nucleus. So what does that mean? That we have the hypothalamus somewhere over here in the middle of the brain, and then right above that, the optic nerves from our eyes, they move backwards and then they cross over. That crossover of the optic nerve pathways is called chiasm. So just above the chiasm, the hypothalamus, there is a structure called hypothalamus, which is below and above the chiasm as well. So just above the chiasm of the optic nerve pathways is the part of the hypothalamus that produces vasoactive intestinal peptide. Similarly, in our GIT, pancreas produces VIP, and then the GIT itself can produce VIP as well, which in turn, for example, vasoactive intestinal peptide can then work on the chief cells and cause pepsinogen release, and it also reduces the gastric acids and so on. So we'll talk about those, but it has a very important role in the GIT as well. In addition to that, it has roles in the respiratory system too. So in our discussion today, keep two roles in the respiratory system of the VIP in your mind. There is one bigger role, and that is, and by bigger I mean a macro function, and that is that the bigger airways, central airways, that means the tracheas and the bronchi, these central airways are dilated by VIP. So that is a bigger mechanical function in the lungs. And then on a microscopic level, it also works on the type 2 pneumocytes, and we'll talk about that in a second. It also works on them and causes them to, I'll describe that it protects them. So two roles in the lungs. The second role, where it protects the pneumocytes, is more important for us today. Another third system today that we'll talk about is the immune system. So keep in mind as well the function of VIP and then Aviptadil on the immune system. It, in a way, this function is very similar to leronlimab-like function, plus more. So there are a couple of things over there. So continuing, what are the functions? So let's start from the GIT. And again, the discussion today is more focused on the COVID. But I wanted to make sure that we understand what is VIP and what is Aviptadil in general. So VIP, or vasoactive intestinal peptide, when it works on the GIT, the basic function of this is to actually increase the GIT motility. That means the movement of it. And please remember this, whenever we cause our GIT to move faster, we try to open the sphincters as well. You do not want the GIT to move very fast and then the anal sphincter is not open, and now you'll have cramps and pain. Similarly, the esophageal sphincters relax as well when the GIT motility start, so that if we have to vomit, we can vomit. So normally the sphincters relax when the other muscles in the GIT move faster. And when the other muscles in the GIT move slower, then the sphincters contract. So that is the general idea. Plus, also remember that whenever we have increased motility, usually with that we have increased secretions produced from the GIT as well, because now we are in a hyperfunctioning mode. So if you keep that in mind and look at the function here, you would see that number one, it relaxes the VIP, the vasoactive intestinal peptide. It relaxes the lower esophageal sphincter. So the lower esophageal sphincter somewhere over here is relaxed. Stomach is relaxed. Gallbladder is relaxed. So that is one. It also, the VIP also increases the secretions of water in pancreatic juice, bile, and it also increases the pepsinogen production by the T cells. So secretions are increased in general. Then it reduces gastric acid secretion, and it reduces the absorption of stuff from the GIT. And it's a very simple thing. Look, whenever the GIT would move fast, it is going to move food and the substances in it faster forward or maybe backwards. And that would actually cause them to be ejected from the body fast. So the absorption would naturally reduce. So the VIP reduces the gastric secretion, plus it reduces the absorption of the content from the GIT. On the heart, so this is the beautiful heart I made for you. So on the heart, it causes coronary vasodilation. And that is a general mechanism, if you see, for the VIP, that just like in the lungs, that it causes bronchodilation. How does it do the bronchodilation? It does that by relaxing the smooth muscles. Same is the situation with the smooth muscles in the blood vessels. So the blood vessels of the heart, the coronary arteries, are relaxed and dilated by the VIP. It also has positive inotropic and chronotropic effect on the heart as well. Inotropic is the force of contraction of the heart. Chronotropic, the word chrono in here means time. Chronotropic effect means increased heart rate or increased number of beats in a unit time. So in general, what happens is now the heart's own blood supply is increased, plus heart's force of contraction is increased, plus heart's number of beats per minute are increased. Normally, this can be useful in failing hearts. So then continuing on, in addition to that, so some other functions. Now remember, Aviptadil's primary function for which it is sold is erectile dysfunction. And erectile dysfunction is also a problem with the blood vessel dilatation. So what happens is that when we dilate the blood vessels in the penis, that causes more blood to become accumulated there or engorged there, which causes the penis to erect. And so if somebody has erectile dysfunction, giving drugs that cause the vasorelaxation, that causes more blood to accumulate in the penis, and that causes the erection. So basically, Aviptadil is used for erectile dysfunction. In addition to that, VIP, vasoactive intestinal peptide, which Aviptadil is a synthetic peptide, correct? It also doubles the vaginal lubrication. So in the presence of VIP, the normal lubrication is doubled. It also regulates the prolactin secretion from the higher centers of pituitary. Prolactin is used for milk production. So normally what happens is women's body, actually both men and women, they produce, or their higher centers try to produce prolactin all the time. So they try to produce milk all the time. However, there is a prolactin inhibition by the still higher system, for example, hypothalamus, on the pituitary to suppress the milk production. Men normally cannot produce milk. That is not because they do not have prolactin. That is because they do not have breasts. They do not have glands that are developed to produce milk. Women can produce milk because their glands are developed. However, women who do not have children or who do not have babies or fetus, they do not produce milk because their higher center, hypothalamus, is usually keeping the prolactin level suppressed. But when a woman becomes pregnant, then the suppression of the prolactin is removed. Then prolactin goes to the breast tissue and starts with the milk production. Milk production can be actually done in many other ways as well. For example, stimulation of the nipple can cause milk production. Some mothers can actually just look at the baby and they start having the milk production. So there are many other ways as well that the milk production can happen, but prolactin is the hormone which works with that. Now, we are now going to go to the microscopic level. So this is the important part. And yes, Luffy just started howling. He was totally quiet before I started speaking. So I think he always wants to have a debate with me to say, okay, I'm going to talk as well. So here, pneumocytes. So in our respiratory system, especially in our alveoli, so if I make the respiratory system here, and I'm going to ask you this thing in a second. So let's say this is the bigger airways, so trachea and the bronchi. From these bigger airways, we get lots of branches, and we keep making smaller and smaller branches or new generations of the branches to the point that finally we have very tiny, small, sort of microscopic airways. And then at the end of them are these sac-like structures which are called alveoli. The gas exchange occurs in these alveoli, this area. This is where the oxygen, when it comes in, goes through the blood from here, and carbon dioxide from the blood goes back into the alveoli. And we have done this discussion in our past lectures, that this part here, the epithelium of the alveolus, the connective tissue here, and then the endothelium of the blood vessel, these three things together make the respiratory barrier. This is the barrier that has to be crossed by oxygen and carbon dioxide, oxygen to go into the blood and carbon dioxide from the blood to go back out. And please remember, now that we have drawn this, Aviptadil actually helps maintain this barrier, and coronavirus tries to destroy this barrier. And how does it do that? It does it by, and we will look at those cells in a second, in the alveoli. There are two kinds of cells. We have flat cells here, which are called pneumocyte 1 type 1 pneumocytes. And then we have tall cells here that are called type 2 pneumocytes. VIP or Aviptadil is going to, just keep this in mind at this time, they are going to work on type 2 pneumocyte. Why would it do that? Let's just talk about it. So if I go back here, so this thing that you're seeing is that respiratory barrier that I just showed you. This is the alveolus, the last terminal part of the airways. This is the connective tissue, this green one. And then this is a capillary in which there is blood. Now what happens is oxygen will arrive in this area, and then from here it would cross the pneumocytes or the cells of the alveolus. Then it would cross the connective tissue between the alveolus and the blood capillary. Then it would cross the capillary endothelial cells, and then it would enter the blood plasma, and then from there RBC, inside the RBC it would go to hemoglobin and get attached there. Now check this out. Type 2 pneumocytes, these tall cells here that are saying you hi, they have two interesting receptors on them. Number one, they have a lots of ACE2 receptors on them. which means what? That will mean coronavirus will attack them very often. Secondly, they also have VPAC receptor on them, or VIP1 receptor. So this is the receptor where vasoactive intestinal peptide will come and connect. So you can think of it for the time being as ACE2 will be connected with the coronavirus, and then coronavirus would use that enzyme to try to cause damage to us by going into our system, and we know all the mechanism. On the other hand, on the same cell, there is VPAC1 receptor on which VIP can attach and try to defend against the ACE2 receptor's behavior. So you can imagine that one of this, ACE2, is for destruction in context of coronavirus, and VPAC1 is for protection. And for the protection, the enzyme that we need or the peptide that we need is VIP, or the drug that we would need from outside is going to be Aviptadil. So now let's see what is going on here. Type 1 pneumocytes are these flat cells. They are 95% of the alveolus. Type 2 pneumocytes are these tall cells. They make 5% of the alveolus, but check this out. Type 2 pneumocytes are responsible to make type 1 pneumocytes. So type 1 pneumocytes are like the children of type 2 pneumocytes. So type 2 pneumocytes are parents. It is their function to replenish type 1 pneumocytes. So whenever, for example, if somebody is smoking and that smoke is causing damage to type 1 pneumocyte, or somebody has coronavirus and that is destroying the type 1 pneumocytes, the type 2 pneumocytes function is to build and make more type 1 pneumocytes. Not build, but actually divide and make more type 1 pneumocytes. So this is one function of that. This is a stem cell-like function, but type 2 pneumocytes do other things too. What other things they do, which are really important, and that is they make surfactant as well. Surfactant is a fluid layer or water layer. It's not exactly water. It is fluid layer that is inside the alveoli. The function of this fluid layer is to reduce the surface tension within the alveoli and prevent them from collapsing. So this surfactant production is by the type 2 pneumocytes. Then, of course, they take part in oxygen transfer. That simply means that the oxygen passes through them, which is fine. It passes through type 1 pneumocytes as well. And then they produce type 1 pneumocyte production as well. So these are the functions of type 2. Now, considering that these are the functions of type 2 pneumocyte, you can imagine that if a type 2 pneumocyte is destroyed, then not only that part of the lung is damaged, but the capacity to fix that part is also damaged, because type 2 pneumocytes function is to fix that part by producing more type 1 pneumocytes. This is why it is important for us to protect our type 2 pneumocytes. And guess who would help protect them? VIP. So this is why we say that VIP, or vasoactive intestinal peptide, or the synthetic thing is the Aviptadil, that has respiratory barrier protective function. So hopefully this much is clear. So at a macro level, bigger level, it causes bronchodilation. And at a microscopic level, it helps protect the respiratory barrier. And I think I forgot to talk about one thing over here. Maybe it is next. No, so it has to be here. 1 second, please. Yeah, here. So I forgot to talk about this thing over here. This is the VIP is released in our lungs as well. And when it is released, when it is secreted in the lung, it is secreted by autonomic nervous system. Our autonomic nervous system, or the nervous system that is automatically controlling our visceras and our functions without our desire. So for example, heart rate goes up or down, or the GIT moves fast or slow, or the heart goes fast or slow. All of those things are automatically controlled. Breathing rate is automatically controlled. Blood pressure is automatically controlled. So the nervous system that is controlling them is called autonomic nervous system. Autonomic nervous system normally produces two kinds of neurotransmitters. One is called cholinergic type, and the other one are called adrenergic type. However, VIP is also a type of neurotransmitter that is released by autonomic nervous system. So all those neurotransmitters that are released by autonomic nervous system, which are not cholinergic or adrenergic in nature, they are all called non-adrenergic, non-cholinergic autonomic nervous system transmitters. Because of that, we say that VIP or vasoactive intestinal peptide is released by NANC nervous system. So what is NANC nervous system? It is nothing but non-adrenergic, non-cholinergic nervous system. So I wanted to make sure that and usually this VIP is inhibitory. Relaxation of the bronchi is an inhibitory function. We stop the smooth muscles from contracting, we inhibit them. That is how the bronchi relax. The vasodilatation is an inhibitory function. We stop the smooth muscles of the blood vessels to contract or from contracting, and the blood vessels relax. So that is a inhibitory function. So VIP acts as an inhibitory neurotransmitter. So there's a question here from Guy that autonomic control by hypothalamus. That's correct. So now going back here, I had missed that one point, so I wanted to do it. So back here now. Now we're going to focus on this little pneumocyte, type 2 pneumocyte. That is this pneumocyte, this guy. Now let's look at him a little bit more close. So type 2 pneumocyte on one end has ACE2 enzyme on it. And if you see here, I know that cool beans are now very much aware of what is the structure of attachment of coronavirus. So remember, there is this TMPRSS2 protein, which is blocked by bromhexine and by camostat mesylate. This protein would prime the S protein of the coronavirus. Then the S protein of the coronavirus would connect with ACE2 enzyme. A part of that would also connect with the GM1, and we talked about the bromhexine as well. So the GM1 is also or ganglioside type 1. So this is the attachment of the coronavirus. The result of that attachment is going to be that coronavirus would end up in the cell and start making more viruses and kill the cell in the process. Now what happens is, inside the cell, when the cell is discovered, a sick cell is discovered by natural killer cells or CD8 cells, they would instruct this cell to kill itself. And that is a suicide by a cell. And it's a normal, natural process. However, the process of suicide is done by proteins called caspases. I always call them the friendly, the Casper, the friendly ghost. So the caspases proteins will order the cell to kill itself. So what happens is, on this side, you are seeing Aviptadil. It is hugging and kissing the pneumocyte. It is protective for type 2 pneumocyte. So it connects with the VIP1 receptor on the same pneumocyte, and then when it connects there, it blocks. So the secondary mechanism inside the cell would result into a blockade of response to caspases. So now the cell is telling the virus has come in and the cell is sick, and the natural killer cell came over and told this cell to kill itself. But in the presence of VIP, the cell is going to say, I'm sorry, I'm not going to kill myself. And this protective mechanism is very important. And it is not only for the type 2 pneumocyte, it is for the immune system cells as well. But this protection, how does that help? Number one, a cell that is protected from dying would stay there and continue to function. Number two, when a cell will not die, the broken down products of the cell will not be detected by the immune system, and there will be less inflammation, and there will be less fibrosis, and there will be less tissue damage. So if we can make the current cells present, if we can make them strong and say, you stay strong and you stand here and don't die, that would actually reduce the fibrosis and reduce the scarring and reduce the damage. So this is where Aviptadil is really, really useful. In addition to that, so it does many things over here. So number one, it causes the caspases blockade. It also causes on the immune system cells to not release tumor necrosis factor alpha and IL-6, and we'll talk about that a little more. In addition to this, there is one more very important function. and that is that Aviptadil or VIP causes reduction in edema that is caused by HCl. So when the damage starts occurring in the lung, there will be HCl produced there, which in turn would cause edema production, and that is acute respiratory distress syndrome. So it actually reduces the piling up of fluids in this area as well. All of this is what we say it is protective for respiratory barrier. It will try that the type 2 pneumocytes do not die. It would try that the immune system does not overreact. It would try that extra fluids do not accumulate in the lungs, and so the lungs stay protected. Then now to our favorite diagram on the immune system. And I want to show you this, this document here. I have the link to this document in the description. It is a beautiful document, and I believe it's 130-40 pages. So going over all of it is difficult during the lecture, but I have gone over the majority of this. So what I read about was, how does it work? How does VIP or Aviptadil work with the immune system? So that's what I'm going to explain. This is the discussion that is here. So let's look at it. Number one, so this is a dendritic cell. So this could be a dendritic cell, this could be a macrophage, this is an antigen presenting cell. And then, so there is a side discussion going on that is going to be $10 very quickly. Oh, wow. Okay, so there's a stock discussion going on. So you guys have your own discussions, and I am left out just talking about the lecture. You should include me in your discussions too. All right, so here, this is a dendritic cell, correct? Dendritic cell, once again, look at it. It has the ACE2 enzyme for the coronavirus to come and attach, although that was not the function of the ACE2 enzyme, but oh well, that is how it happens. Plus, dendritic cells with the macrophages have toll-like receptors and PAMPs and the other receptors with which they catch the pathogen. So once the pathogen is caught, we know that that pathogen will then be presented to naive T cell. Then in the presence of interleukin-4, there will be T helper 2 pathway taken, which would then cause antibody production, and this is called humoral pathway. Or in some cases, T helper 1 pathway can be taken, which in the and that happens in the presence of interleukin-12, which would then cause cytotoxic T cell production to occur, which would cause cell death. And we prefer T helper 2 pathway over T helper 1 pathway in case of coronavirus, because T helper 2 pathway is anti-inflammatory and T helper 1 pathway is pro-inflammatory. We also like to have T regulatory cells because they balance the immune system, they reduce the immune system's reaction. So now let's look at how VIP or Aviptadil function in all of this. Number one, on the macrophages and the dendritic cells. On them, there is a receptor for the VIP, or called VIP1 receptor. So there are 3 receptors for VIP or Aviptadil, called VIP1, VIP2, and VIP3. So VIP1 type receptor is present here. When the VIP attaches to that, that causes reduction in chemokine productions. Now this should remind you of the leronlimab as well, because CCL5 and RANTase, that is where the function of the leronlimab comes in, which disrupts this axis. What is the function of the CCL5 and RANTase? Remember that when CCL5 or RANTase is produced, that causes the immune cells, for example, macrophages, monocytes, T cells, to gather in that area. It is kind of a remote control to say, hey guys, come over. And leronlimab disrupts that, that behavior. This is a similar behavior that can also be done by Aviptadil. Again, there is a clinical trial. There is nothing yet done, and well, that's the case for leronlimab as well, that there is a trial being done, but the results are still being waited on. So CCL5 and RANTase production is reduced. That is a leronlimab-like function. CCL4 production is reduced. Very good. CCL3 production is reduced. These all 3, when their production is reduced, that reduces the bringing of the macrophages, monocytes, and the T cells to the area of infection. So that is good. Then CCL1 and CCL2 production, CXCL1 1 and CXCL2 production is reduced as well, which causes less neutrophil to come to the area of infection as well. So that is also good. So now VIP or Aviptadil is causing many functions over here. Remember, it has caused bronchodilation. Remember, it has protected the type 2 pneumocytes too. Now it is actually protecting the immune system as well, or helping the immune system too. So there is a bigger function that is the result. Now, if you go over here, this is the naive T cell. Naive T cell has VIP1 receptors on it and VIP2 receptors on it. VIP2 receptors can normally be activated, and if they are activated by VIP, that would cause the naive cell to go T helper 2 pathway. So that means giving Aviptadil would take the system towards T helper 2 pathway, which may be actually a good thing because it is less inflammatory. When a naive T cell becomes connected with an antigen presenting cell, then the VIP2 receptor on it also becomes active. Did I say VIP2 before? VIP1 receptor also becomes active, and then in the presence of the VIP1 receptor, and if there is VIP or Aviptadil present, then the function of the naive cell would go down. So that is another I would not call it immunosuppressant, I would call it immunobalancing function. On the antigen presenting cell, this is another interesting thing, that when the antigen presenting cell has connected with the naive T cell to start the adaptive arm of the immune system, at that time, if VIP is present or Aviptadil is present, then the co-stimulation, remember we talked about it, that the hugs and kisses occur between the cells and they co-stimulate each other, and that is how the cells become active. The co-stimulatory molecules and their expression is reduced on the antigen presenting cells. So they do not activate too many of the T cells. Good, that's a good thing. So they're not calling more cells over there. They're not activating too much of the adaptive arm. Adaptive arm itself is also protected by VIP and or the Aviptadil, because they are responding in the right pathway and they're responding by responding less. Then in the presence of VIP, the pathway taken is T helper 2. Then over here on the B cell side, VIP receptors are also present, and they also modulate the B cell response as well, or antibody production response too. Very good. In the presence of VIP, the naive T cell can also go to the T regulatory cell, which is an immunosuppressant pathway or immunobalancing pathway. So once again, the cytokine storm will not occur, or hypothetically will not occur. Then thirdly, in the presence of VIP, because we are taking T helper 2 pathway, we will not take T helper 1 pathway, which is pro-inflammatory. Then the cytotoxic T cells have VIP1 receptors on them as well, which help them become active, and it helps them reduce the production of tumor necrosis factors and other cytokine. So tumor necrosis factor production is reduced. Interleukin 6 production. Remember interleukin 6? We have other drugs for interleukin 6 as well. So that production is reduced. The Rantese production is reduced. The immune pathway, correct pathway is taken. Cells' overwhelming response is controlled. And then overall immune system becomes balanced. In addition to that, there is one more quick thing. I have it over here. So we saw that co-stimulation is reduced. Very good. T helper 2 cells plus CD8 cell produce VIP as well. They themselves can also produce vasoactive intestinal peptide. CD4 cells become active with it. We know that T helper 2 pathway is taken, T helper 1 pathway is suppressed, and T regulatory pathway is taken as well. That is the effect of this thing on the immune system. In my opinion, if it works, it is actually going to be a very powerful drug. Now, let me show you a few more things. It has been in production for 20 years. And for the 20 years, it has been going through the trials. So phase 2 trial, it has for sarcoid, pulmonary fibrosis. So can you tell me why pulmonary fibrosis? We just talked about it. It is protective for the pneumocytes. So when they're not destroyed, the fibrosis production is less. It is protective for the immune system as well. So when immune system is not overreacting, then less damage and less fibrosis would occur. Bronchospasm, and that is where bronchodilator effect of the Aviptadil would come in. So it has been going through the phase 2 for these diseases. And now for acute respiratory distress syndrome, which is kind of more relevant to COVID-19, that it is going through the phase 1. And in the phase 1 of the Aviptadil for acute respiratory distress syndrome, there were 8 patients taken. Those patients were given IV Aviptadil, I believe, and then they were all on ventilators. They were all intubated. After giving this drug, 7 of them were extubated within, I think, 4 or 5 days. 1 person had died. Now, usually it is given in the inhaled form. The reason for it to give in inhaled form is that when you give Aviptadil in systemic form, it has many effects in lots of parts of the body, and so side effects are more. But when you give it inhaled, then it goes and works primarily on the lungs and helps with the lung. However, I'm going to show you a clinical trial, and for that clinical trial for COVID-19, the drug that they're giving, Aviptadil, is going to be IV. So that is the discussion of Aviptadil. Let me now just very quickly show you a couple of links over here. This link is Vasoactive intestinal peptide, or VIP, inhaled agonists, potential role in respiratory therapeutics. So that is one. It's an interesting read, so please feel free to do it. This is about the Aviptadil on Wikipedia, not very detailed here. Then this is the VIP, vasoactive intestinal peptide. This is a larger topic here, and I just discussed it. This is the clinical trial for Aviptadil. And if you see here in this trial, they're saying that Aviptadil, a synthetic form of human vasoactive intestinal peptide, has been awarded FDA orphan drug designation for the treatment of ARDS and admitted to the FDA Coronavirus Technology Accelerator program. So that is one. Secondly, they're talking about the VIP itself. They're saying that the VIP, it prevents NMDA-induced caspases. We talked about the Casper the ghost. It prevents production of interleukin-6 tumor necrosis factor production. And we actually saw that it's much more than just these two. It protects the pulmonary edema development because of HCl. It upregulates the surfactant production by the type 2 pneumocytes. So the whole respiratory barrier becomes protected. Then Aviptadil has a demonstrated, and we talked about it, that phase 1 and 2. Down here, if you see, they've started this study since May 15. They're supposed to finish it in August, and September we'll have results. So very soon we'll know what is going on there. Then down here, there is a dosage here, which I thought was interesting. So here's a dosage. Experimental Aviptadil IV, not inhaled, in escalating doses, plus the rest of the care. So what are the dosage? Patients will be administered Aviptadil IV in escalating doses of 50 picomole, 100 picomole, and 150 picomole per kilogram per hour. And then the other part of the treatment is normal care. So it will be interesting to see what is the result of this one. This is the discussion for today. From a mechanism point of view, I feel very excited about it. It has the function of interleukin-6 blockers. It has the function of leronlimab-like drugs, and it has further functions as well. So looks promising. We'll see how it goes. So thank you very much, and see you tomorrow. Please do me a favor, like, subscribe, and share. So love you all. Stay safe and healthy, and I will see you tomorrow. Bye-bye.